Okay, so over the last couple of lessons, we've been talking about protein synthesis. So just to quickly recap, we've been looking at DNA as a double-stranded molecule with the term DNA standing for deoxyribonucleic acid. So the deoxyribo part is the sugar and it is a nucleic acid. So the whole strand is used for a template when we are undergoing DNA replication. And the bases that are involved are adenine, thymine, cytosine, and guanine. RNA, on the other hand, is a single-stranded molecule with the letters RNA standing for ribonucleic acid. So as you can see, RNA is also a nucleic acid. However, the sugar in this case is simply ribose instead of the deoxyribose. Only part of this strand is used as a template, and the bases we have adenine, cytosine, guanine, and uracil. So when there is an A on the DNA strand, the mRNA strand that results has a U instead of a T. So looking at this picture, we can see we have our double-stranded DNA and our single-stranded RNA, very similar in structure. Simply the differences being DNA is double-stranded, RNA is single-stranded. DNA has C, G, A, and T, whereas the RNA has C, G, A, and U. So we have three different types of RNA that we need to know about. In particular, we need to know specifically about mRNA and tRNA. So mRNA, M standing for messenger, is copied straight from the DNA template and can be up to hundreds to thousands of nucleotides long. So we use the DNA in the nucleus as a template that is then transcribed into the mRNA. Transfer RNA or tRNA is also copied from the DNA template and is only 75 to 85 nucleotides long, so much shorter segments of RNA, and that is used during the translation phase of protein synthesis. We also have ribosomal RNA or rRNA, which help to make the proteins within the ribosome. So the ribosome is the organelle in the cytoplasm that is involved in protein synthesis. So for protein synthesis to take place, the DNA unzips to make the code or message for the unit of protein known as a polypeptide. The DNA unzips and one section of the strand acts as a template, so that is referred to as a gene. So the gene is transcribed into a single strand of messenger RNA or mRNA. That is then carried from the nucleus to the ribosomes in the cytoplasm, and transcription of DNA is followed by translation. So the creation of the mRNA is known as transcription, and it occurs in the nucleus. So we talked about the fact that transcription and nucleus both have a C, so that's one way of remembering that transcription takes place in the nucleus. So the formation of mRNA uses the bases of the DNA code as the template. So transcription occurs mainly in the nucleus, but it can also occur in the mitochondria or the chloroplasts. So if you recall back to year 11, these are the two other organelles that have their own DNA. However, majority of the time it takes place in the nucleus. And the enzyme that is used to separate the two strands of DNA is RNA polymerase. Translation is the second stage that occurs in protein synthesis. So transcription takes place in the nucleus, creates the mRNA. The mRNA then moves out of the nucleus through the nuclear pores into the cytoplasm, and it attaches itself to a ribosome. So a ribosome is a circular organelle within the cytoplasm. The tRNA also leaves the nucleus via the nuclear pores and will attach itself to an amino acid, which is also in the cytoplasm. So the function of the mRNA is to transport the code of DNA from the nucleus to the ribosome, where it is then translated. So the way that this happens is as the mRNA attaches to the ribosome, it is read in a series of three bases, which is known as a codon. So those three bases act as the template or the structure for the tRNA. The tRNA then matches the appropriate codon with its own anticodon, and the other end of the tRNA has an amino acid attached. So we have 20 amino acids, however there can be 64 different codons. So some amino acids have more than one codon that codes for that particular amino acid. So the tRNA brings the appropriate amino acid in, the codon matches with the anticodon. Then when the second tRNA brings the second amino acid in, the two amino acids join together, and then the first tRNA goes on its way to find another amino acid, and then the cycle continues until we have a series of amino acids being bonded together with a peptide bond to create a polypeptide. So these polypeptides then become the proteins. So as we can see in this picture, it summarizes it quite nicely. So inside the nucleus, we have our DNA, which undergoes a process of transcription to form mRNA. The mRNA then leaves the nucleus through the nuclear pores and enters the cytoplasm, where it attaches itself to a ribosome to undergo the process of translation. During translation, the codons are read as they pass through the ribosome, and the tRNA with the matching anticodon carries the amino acid, and then those amino acids join together, and we have the process of protein synthesis taking place. In the slides, there's a really good video that you can watch. It only goes for a couple of minutes that explains how protein synthesis takes place with some really nice visuals, which you will find here. Okay, thanks guys.